in my lab and Gordon Lithgow's lab at the Buck Institute, PDL Health. This is disclosure. I'm involved with the company, so now you know. The goal was to try to find natural products that would have additive effects on lifespan and health span. And I think there were two reasons for this. One is that we thought that hitting multiple pathways, especially with natural products, would likely have a bigger effect than any one pathway when we started this project. And of course, there was a practical reason too, which is that to generate IP in the natural product space, you need to have combinations of different natural products or different formulations, et cetera. So we were trying to achieve both goals. And the molecule that gave us the biggest effect out of a screen of about 40 different natural products that had been linked to aging in one way or another was alpha-ketoglutarate. And so the initial studies were done in worms, looking for compounds that worked and had additive effects. But this is the first study on alpha-ketoglutarate many years ago now, published in Nature, showing quite a big effect of this metabolite on longevity in C. elegans. And we're able to repeat this effect, but I'm not going to show you our worm data today for the sake of time. That was mostly performed in Gordon's lab. I'll show you the mouse data, which we did a lot in our lab. And this was a graduate student, Azar, that worked a lot on this. She just got her PhD. And so she was co-trained by Gordon and I. So we looked for, when we switched to mouse studies to validate what we found in worms, we started interventions at 18 months. This is kind of following the original rapamycin study, showing that a relatively late intervention could still have an effect on aging. And given that this is around the time we want to do clinical studies, we thought that might be a good time for the intervention in mice as well. Now, if you just looked at this slide, you wouldn't see anything to get you extremely excited. There is an effect on lifespan. There's about a 10% extension in females, 5% in males. This is cohort 1. We've done a second cohort and got the same effect. But that's a relatively modest effect. The thing that really jumps out about alpha-ketoglutarate is its effect on health span and frailty. And so we're using Susan Howlett's frailty index, this 31-point index that was published several years ago that's becoming more popular. We measured frailty either in the control animals or the alpha-ketoglutarate treated animals. The controls are in blue and the AKG is in magenta. And you can see as the months go by, the control animals get more frail than the AKG treated animals. But this is a very complicated way of presenting this data because we're not accounting really for the animals that are dying along the way. By the time you get to 2 years of age, some of the animals are beginning to die. And for instance, this point on the controls here, this animal has a very high frailty, but is not present in the 25-month time point because the animal's dead. So after all the animals dead, we went back and recalibrated this by calculating at what point during the animal's lifespan we measured the frailty, either between 50 and 60%, 60 and 70, 70 and 80, 80 and 90, and 90 and 100. And what you can see is you get a much more linear response, the frailty response in the control animals. It goes up steadily with age. And it's strikingly reduced in the AKG-treated animals. There's about a 50% reduction in females. I'm not showing males, but there's about a 40 to 45% reduction in males. So you see almost equivalent effects on frailty in both sexes. And so what we would like to argue from this is that this natural product or metabolite is compressing morbidity. It's keeping the animals healthy very late in life and also extending lifespan, but the frailty effect is bigger than the lifespan effect. Another way of plotting this is to take all of the frailty measures and plot versus the remaining lifespan of the animal. So we've ignored the longitudinal component here and just plotted all the data points. And what you can see using this frailty index in blue is in the control animals, frailty accurately predicts mortality, both in the female and male populations. But the AKG animals, it barely predicts mortality in the female animals and really has no significant prediction in the males, again, indicating that the frailty is largely suppressed in the AKG-treated animals. So what is AKG? You probably should know if you took a biochemistry class at some point. this central metabolite in the TCA cycle. And it's a little bit like NAD in the sense that it's also a substrate in hundreds of different chemical reactions in the cell. And so we see an effect of AKG, but that doesn't lead to an obvious mechanism. And there are many different potential mechanisms by which it can be affecting aging. And in fact, we've shown that it's associated with improved adult stem cell function. We think it improves metabolic flexibility. In some contexts, we can see enhanced respiration. There are alterations in the microbiome that we're just beginning to study. There's a relatively robust effect on inflammation, and that's something I'll show one slide on. And in the human studies, the company is already the product's already available, and we're getting feedback from people taking it. We are seeing changes in the epigenetic clock, but that's not in a clinically controlled study, which those are ongoing. I can tell you about them in a second. Just briefly, one of the big effects we saw was on coat condition and hair growth. And we were able to show that when you treat the animals with alpha-ketoglutarate, the coat color may be explained by the fact that you preserve the melanocytes in the hair bulbs much later in life in the AKG-treated animals than the control animals. So that we see a big effect on some of the aspects of frailty, and the ones involved in coat condition and kyphosis particularly are heavily affected. Here's the inflammatory effect. These are all inflammatory cytokines. Starting 18-month control animals, it's all set to a neutral color, and you can see that the increased red in the control animals at 28 months shows increased inflammation. And that's largely suppressed in the AKG-treated animals. I want to jump to something else. Most of that data is in the paper coming out. I want to jump to something else, and that's the AKG and exercise performance. Several people that are taking this are reporting to us that they have better exercise endurance. Again, anecdotally. But that's not particularly surprising because there's a long literature on alpha-ketoglutarate and muscle building and exercise performance. The supplement may enhance athletic performance and metabolism. These are just 3 papers I pulled off the web. There are many others. It's used in increasing cell growth and healing and injuries and wounds, and particularly important in healing of muscle tissue. A non-essential amino acid that may be beneficial in muscle building efforts of exercising. And so this has been in the literature for a long time, but the mechanism's not entirely clear. Now, many of the supplements bodybuilders take not only have AKG, but they have amino acids like arginine in them as well. And I just want to be clear that we're only giving AKG in these studies I'm showing you, and not the amino acids, which may have other effects on aging we're not sure of. And let me go back. One particular challenge in trying to understand mechanism is when we give AKG to the mice, or when we put it on cells in cell culture, we found, and this was already known, we rediscovered that AKG doesn't get in easily to cells. So it's a molecule doesn't cross membranes easily, and they're not necessarily mechanisms to drive AKG into cells. And so many cell types, when you add AKG, it's not getting in at a very high level. There is, when we give it to mice, we see it get into the intestine and the liver, but a lot of peripheral tissues, we can't detect an increase in the labeled AKG. One place, though, where we forgot to look in our first study was red blood cells. And this paper actually was published before I was born in 1963, talking about utilization of alpha-ketoglutarate in red blood cells for glutathione synthesis. So red blood cells, as you know, are quite different. They don't have mitochondrial function, and so when they still need to make molecules like glutathione. In fact, glutathione is particularly important in red blood cells because they're carrying lots of oxygen, and many molecules can be damaged by oxidative reactions, including hemoglobin itself. And what's alpha-ketoglutarate is one of the principal sources of the glutathione production in red blood cells. And in fact, there have been papers out showing that AKG is a better substrate to be converted into glutamate for glutathione synthesis than glutamine is. And in fact, in some studies, up to 89% of the glutamate in glutathione comes from AKG in red blood cells. So one thing we think we're doing is increasing glutathione synthesis, and we can show that. So this is total red blood cells here, where we're adding at time 0 AKG, and what we can find is the red blood cells do take it up, and you see an increase in glutathione levels over a 2-hour period. In mice treated for a week with AKG, you can see increased glutathione levels both in males and females. This is a very small number of mice. Now that COVID-19, we're back in the lab after COVID-19, we're trying to make this data more robust, but I think it's very suggestive that we're increasing glutathione function. So in my view, this may be one of the reasons you see better exercise endurance when you're taking AKG, is because you get better red blood cell function and better oxygen delivery to muscle cells. So that's one of the mechanisms that may be affecting aging as well. So just in the latter part of one more slide, sorry. So we have 2 human clinical trials, one going and one planned. The one going is sponsored by the company, and it's being done at Indiana University. The idea is to do a 9-month intervention up to 100 people, 45 to 65-year-old participants, placebo-controlled, looking at aging biomarkers and inflammatory markers. And there's a similar study that we're planning in US here in Singapore, except that we're only going to use AKG. The product has some vitamins in it as well. And in this study, what we would like to know are specific mechanisms that are being affected by AKG, so we're only using that. Otherwise, it's a similar study. So hopefully we'll have some data on that in the near future if we can get these clinical trials up and running after COVID. I want to comment on validating longevity interventions, though, because we're all kind of struggling at this point with how do we take interventions we think are going to affect aging and test them in humans. And there have been 3 major approaches that have been taken by this of aging-based companies over the years. One of which is to really take the small molecules that hit longevity pathways and then try to treat disease or complications of disease with them. This makes a lot of sense if you have a new chemical entity or you're a pharmaceutical company, because you need to get reimbursement and FDA approval by the standard paradigm through these companies' work. So if aging is not a disease, you can't get reimbursed or approved to treat aging. So I get that. But there's been several high-profile failures, I think, including a couple recently, using this approach. And I want to make the argument that while this is practical from a new chemical or a new drug point of view and from a standard pharmaceutical point of view, that it may not be the best way to move these aging interventions effectively into humans. And I think we need to give serious thought to how we're going about doing this. Now, there's the other approach, which is to prevent disease. And so the TAME trial with near Barzilai would be a great example of that. Give the metformin, in this case, to people in their 60s, look at a whole range of different chronic diseases and try to prevent them simultaneously. So this health span type trial is certainly a reasonable approach, and I'm very supportive of it. The problem, of course, is it's expensive and a very long-term study with 14 clinical sites in the US. So it's impractical to test many different kinds of interventions that way. And I think this third approach, which is to use these relatively new aging biomarkers as endpoints, I think is the way that we're thinking of going, and I think we should consider this more. And you're starting to see studies like this that are suggestive of positive effects. And so the idea here is that, especially for lifestyle interventions and natural products, you could do interventions in people, maybe with smaller numbers of people, and then use biomarkers like the epigenetic clock to validate that you're having effect on slowing or reversing aspects of aging. And I want to just make this point one more time. You know, what we're trying to do here is not just an extension of standard medicine. We're trying to go from sick care to health care. And this is really kind of a revolution. We're trying to change how we think about medicine and how to do it effectively, both from an economic standpoint and from improving quality of life. And maybe the best way to do it is not to follow the standard mechanisms of getting things approved and getting things reimbursed, even though that's the route that is the easiest one to raise money for. I think we need to think about the things that are most likely to work. And I think prevention is more where these interventions are likely to work than treatment of diseases or complications. So anyway, that's my 2 cents on the subject. And when you look at these aging biomarkers, you know, they're really rapidly developing. This is from a review. It's actually out now in the journal Proteomics. 10 years ago, I think we were, some of us recognize this, but not everyone in the aging field recognizes this, that the development of interventions has certainly been a revolutionary step in the aging research, but the development of these biomarkers is an equally important revolutionary step, in my opinion, because now that we have these omics-based molecular pattern biomarkers that we can measure biologic age, I think we really have the potential to show that we're affecting human aging. And we can also do the mouse studies now in ways that more easily convert to the human studies. So I think this is a major step forward. None of these biomarkers are fully validated, but I at least believe in a number of them. I think that we're going to find several that are effective. So the first generation approach here was to use estimate biologic age using a single data set and chronologic age. And that led to things like the epigenetic clock, or transcriptional clocks, facial clocks, microbiome-based clocks, and others, metabolome-based clocks. And then more recently, we've seen these second generation metrics, which combine these data sets with clinical biomarkers, and they lead to better predictivity. And I think that these look very, very promising. And I think we need to move to this third generation, which is how do we not only measure the general aspects of aging in the population, but really understand the personalized aspects of aging. And so when I look at it, you have a systemic aging process that's increasing susceptibility to disease. And then you have disease-specific or aging pathway-specific processes going on that dictate which diseases you're likely to get when that aging clock ticks to a high enough number. And I think that we need to start figuring out ways to combine the systemic measures of biologic aging with the disease-specific or the pathway-specific parameters, so that we can not only tell the biologic age of an individual, but what are the things that are likely to go wrong when they get to the point where they can't maintain homeostasis any longer. So in our clinical studies in Singapore, we're using a whole range of different assessments, partly because we want to see how these different clocks, most of the clocks are here, how these clocks correspond to each other, and how they correspond to physiologic measures like body composition, cardiorespiratory fitness, or arterial stiffness, etc. So the one point is to understand, how do all the clocks tell you the same thing? Do they tell you completely different things? I think the data is starting to suggest that they tell you overlapping things. And if that's true, we need to come up with a strategy to get the maximum information in the most cost-effective way, using the right combinations of clocks. And that's really the goal here. And then we'll be testing that with interventions, including exercise, AKG, and hopefully a whole range of other interventions that we can compare and contrast effects on aging in the Singapore population. And so ultimately, our goal is to really build this aging concept in Singapore. Singapore is really ground zero for aging, or it's one of the ground zeros. There's a very long lifespan. Birth rate is 1.1 and per family in the Chinese population, which is 70% of the population. So very low birth rate. It's a small island, not a lot of room for immigration, and so we have to do something to keep people healthy longer. And if we can achieve that, there's huge individual benefits in terms of quality of life. There's also societal impacts, including increasing productive work years, because retirement age is going up. There are government benefits. You don't necessarily have to build as many hospitals and nursing homes, for instance. And we've worked with the Singapore Clinical Research Institute to estimate that there could be a $3 billion savings in healthcare costs if we extend health span by anything up to 5 years. So that's I think, a achievable goal in the not too distant future. And then we'd like to build an industrial component in the biotech space in Singapore as well, and we're working hard to try to achieve that as well. So with that, I think I'll stop and take questions. Hopefully I'm more or less on the right time. The lab's growing in Singapore. The RBC data was by Guan Xiaoping, and the clinical studies were starting or being headed by Zhoming Guo and Jan. And then Chen Yu and Yume and Azar were involved in the AKG studies, along with Garbo back at the Buck Institute. So thank you, and I'm happy to take questions.
Thank you very much, Brian. Really, really stimulating talk. We have a few questions on Slack. And so Here's one from Wilbert Vermeij. He's asking, was EKG affecting nutritional intake, triggering a DR-like response?
We don't see changes in food intake. In some contexts, we see the mice are more active in the cage, although we haven't put them on running wheels and done a lot of work on that. But to the best of, they're not losing weight or anything that you might see with calorie restriction. So we don't think it's mimicking a calorie restriction response.
Very interesting. And we have another one here from Yufei Lin. How was frailty measured in mice?
Yeah, so you can look up papers by Susan Howlett. A number of labs are using this now. They have a couple different frailty indices. We're using the one that's a 31 component measure. It's really mostly subjective measures like coat condition, hair loss, as I mentioned, kyphosis, pilorection, and a whole range of cataracts and a whole range of other things you can measure in mice without being too invasive. What we don't particularly like doing things like micro CT or anything in mice that we're doing survival curves on. So we chose the one that was less invasive, and even though it's subjective, if you do it in a blinded fashion, you have 31 different points that you integrate into this index, and we find it to be pretty reliable.
Yep. I think we're actually also going to hear more about this later in this conference.
Good.
Really nice, Brian. I don't think we have time for more questions. We have our next speaker available, but thank you so much, Brian, for joining. We really appreciate that you were able to join, even though it's...
Anytime. Thanks for arranging it not in the middle of the night, too. That's very helpful.
It's good. Thank you very much.
Thanks a lot.
Thanks. See you, Brian.